Hi, I'm Horace Zhang from the University of Georgia. My talk is about collective discrimination among queens with different supergene genotype by worker fire ants. Animals often need to collectively perform a task as a group, which requires a consensus to be reached among the group members through a process called collective decision-making. Social insect colonies are especially well suited for the study of collective decision-making because they're tightly integrated superorganisms whose survival depends on recurring group decisions. Some good examples involve the nest hunting behavior in Tambothorax acorn ants and honeybee swarming, both cases where relatively few individuals influence the entire colony to move to a new home. Equally important are the decision about which individuals and how many are allowed to become the functional reproductives in the colony, which is the subject of my talk today. So I studied the red and pointed fire ant, Solenopsis invicta, an invasive species common in the southern US. Although most people hate them, they are a good model species to study the collective behavior and its role in social evolution because they're easy to collect and study in the lab. A remarkable feature is that a fire ant colony can have a single reproductive queen, the monogyne form, or multiple reproductive queens, the polygyne form. The genetic mechanism underlying such variation in social organization involve an element termed the SP supergene, which is a large non-recombining section of the genome formed by chromosomal inversions. Ants in monogyne colonies lack this SP supergene, and a queen-like colony won't tolerate any additional reproductive queens. In contrast, all the queens, an average of about 65% of workers in polygyne colonies, have this supergene, and after flight, newly mated queens with the supergene attempt to enter a colony to become new reproductives. Polygyne workers would accept any additional queens that have this supergene or kill those that don't have it. In this way, polygyne workers discriminate in recruiting new queens to replenish the queen pool and grow their colony. Previous studies have shown that as few as 10 to 20% of workers carrying the supergene is enough to induce the entire colony to become polygyne form. But we still don't know exactly how such a low percentage of workers influence the collective decisions about queen number and identity. So here we designed a series of behavior assays to dissect this collective queen discrimination behavior. Hereafter, I'll use M to refer to monogyne and P to refer to polygyne. An assay unit is a colony fragment with 300 workers from a single M colony and 100 workers from 3 P colonies. The control unit is a corresponding M worker fragment with no P workers. Different assays vary with regards to the type and intensity of contact and interaction between the two worker types. We'll first discuss assay A. We set up the M workers in a tray. 24 hours later, we add the P workers. P workers are contained in a cup, and there is a layer of fine metal screen on the lid that prevents the worker from getting through but allows them to antennate ants on the other side. We assess the social form phenotype by introducing 2 unrelated P queens into the unit. In assay A, we release the P workers into the tray 2 hours before introducing the test queens. And if a queen is still alive 48 hours after introduction, we consider this queen accepted. Here the video on the left shows a control unit composed of only M workers, which are attacking the introduced P queens. The video on the right shows a P queen being accepted and retrieved into the nest by workers in the unit with 25% small P workers. So that was assay A. Assay B is similar to A except that the P workers are never released from the cup and thus never freely interact with the M workers or the introduced queens. Assay C further reduces interaction between 2 types of workers by placing a double-layer screen in the cup. The few millimeters of space between the 2 screens prevents the worker from having any physical contact at all. This table provides a comparison of the acceptance rates among the assay types. Overall, as the duration and amount of contact and interaction between the 2 types of workers increases, the polygyne queen acceptance increases. In assay A, where the workers are allowed to freely interact, the results confirm the previous studies showing that a small proportion of supergene carrying workers can convert the social organization of the colony to polygyne form. In assay B, where workers cannot freely interact, the acceptance rate is significantly reduced compared to assay A. In assay C, where the M and P workers have no contact of any type, the colonies effectively behave no differently than the controls. and discriminating strongly against the P queens. Our results suggest that the SB workers may directly influence colony queen preference, perhaps by producing a specific queen retrieval signal when they detect the SB queens. These SB workers may also indirectly influence the colony queen preference by altering other workers' response to the queens. Although a larger sample size will be needed to convincingly show that we can indeed hypothesize such an effect. If the effect is real, it may be due to cuticular compounds on the supergene carrying workers acting as semiochemical signals to manipulate nesmate worker behavior. In this way, supergene carrying workers effectively recruit new queens bearing the supergene while presumably excluding queens that don't have it, driving a consensus colony level collective behavior even if they're only a small minority.